everybody, and welcome. My name is Julie Klingler. I'm the Alumni Director here at the University of Findlay, and it's a pleasure to have everybody back today. We're so happy you're here for lifelong learning. We have had a great month this month, featuring all kinds of sessions, ranging in topics from history to military experiences to medical sessions. And today we've got University of Findlay's Dr. Brian Whitaker, who will join us from our animal science program here at the university. And he's going to talk to us about the important animal science research that he and his students are working on right here on our campus in the Davis Street Labs. So we are going to welcome him in just a moment, but want to just mention a few things for you all tuning in, and we want to make sure that you make the most of this virtual event. So one thing that you can do to help enhance the settings is use the speaker view for the best and largest picture of those who are actually speaking at the time. So make sure you use that. You can adjust that up in the upper right-hand corner of your Zoom screen. And down on the bottom of your Zoom screen, you've got chat feature. Use that to interact with all of your friends and drop your comments in there. We want to hear from you. The Q&A feature is also going to be used today as well. So make sure that you drop questions in there, because we'll have a Q&A session at the end of today's presentation. Make sure also that you're identifying yourself with a really great name, a first and last name, because we use those names for prize giveaways and for chat and Q&A. So make sure that's done. And today at the end of the session, after our Q&A, we will do a prize giveaway. So make sure that you stay with us for that giveaway. You have to be present to win, and we'll do that at the end. But without further ado, I'd like to go ahead and introduce our keynote speaker today, Dr. Brian Whitaker. Dr. Whitaker's been on the faculty here at the university since 2010. He utilizes active learning strategies and student involvement in all of his classes to encourage the development of the whole student. Dr. Whitaker is currently responsible for teaching multiple undergrad courses here at the university in reproduction and swine production and management. He earned his bachelor's degree from the University of Massachusetts, Amherst, and then his master's and Ph.D. degrees from Virginia Tech. So without further ado, I'd like to welcome Dr. Whitaker.
Hello, everyone. I'm so excited to be here, and I'm glad everyone else is here too. So I think as soon as I start to share my screen, we should be good to go. Awesome. All right, so I think the best thing to do is to give you some background information, share some of our facilities with you to begin with, and see what our wonderful marketing program has done here for us today. So we're gonna go ahead and do this.
I chose the University of Findlay because of the reputation they have in animal science and the hands-on opportunities they give freshmen. Being a part of research is very important to me because it gave me a great opportunity to practice my confidence and communication skills and learn more about the animal science industry.
For me, research has been an incredible opportunity where I've gotten to go to multiple conferences all over the US and be able to present my research to a whole lot of people at various education levels and who are maybe more immersed in the research field than I am as a student. I've been doing this for about 2.5 years, and I got to start my own research project, and I've become more confident in my abilities. I've learned how to be a better presenter, learned more about research in general in all areas. And I've even had my research confused with being the research of someone who's getting their master's at multiple conferences. So it's been a very incredible opportunity to be able to talk to different people.
I chose the University of Findlay for my undergraduate program for the outstanding opportunities that the animal science department has, as well as the family-like community that's presented on UF's campus. Research is so important for me, for I've been able to expand my knowledge as well as others within the physiology of reproduction as well as it's also piqued my interest in potentially specializing in reproduction within the veterinary medicine. I chose the university.
So in our lab, we have traditionally been looking at the maturation and fertilization of the pig embryos. And recently we've found, or there's been this big push in society that's a lot of people are waiting to have children, and we're looking at people that are trying to conceive at a much later age. And there's a lot of biology that goes awry with that, because initially the people weren't designed to do that.
It's definitely been a lot of work, but I've also got to learn a lot on the way, as a general about research and what is all involved.
So what we've decided to tackle, or to challenge ourselves, is to look to see if, or during the aging process of the ovary and of the oocytes, the female gamete, if there's some epigenetic modifications that are occurring there.
It's been a learning experience with this research. Prior to joining, I didn't really know the process of research, and so it was really interesting to learn like everything that goes into it, between collecting the data, analyzing it, and presenting it at conferences. I've got to meet a lot of very interesting people through these conferences and hear all about their research, as well as be able to talk to them about my research and what we've done here.
There's definitely an element of problem solving that goes into research, being able to overcome any obstacles that we encounter with a process that isn't working, or we need to figure out what type of chemical to use on an oocyte. So if I do go into medical practice and I come up with a difficult case, that those skills can really help me with overcoming that.
And so I've really learned a lot about the entire process of it and how it helps to further the field of animal science.
The backbone of our research really is the undergraduate students. And a lot of this, most of this wouldn't be able to be accomplished without their help, for the sheer volume of experimental units we go through. All right, so that pretty much just gave you a little bit of some video images of our facilities at the farm, and then of course in our research lab here on campus. I think one of the most common questions I always get from friends, family, people out and about is, why pigs? Of all the animals you could pick, why choose the pig? And you know, there a lot of people either really like them or really don't like them. And early on, I didn't grow up on a farm. I kind of grew up next to a horse farm, but that didn't really count. And no farming background previously, and then kind of just jumped into it in undergrad and said, you know what, pigs are pretty cool. They have lots of really neat things associated with them. They're interactive. And then as the curiosity of biology kind of stoked my interest, I learned a little bit more about them. And one of the things that I think everyone's kind of aware of is that pigs are really a lot like people. If we had to link a non-companion animal, a domesticated animal, to people, it would probably most likely be the pig. Anatomically and physiologically, they are very, very close. They're not a ruminant like our sheep and our goats and our cattle. So digestively and nutrition-wise, the pig is very close to humans. Additionally, if you look at an adult full-grown pig and you looked at the anatomical organs, they're actually the same size as human people or human organs. And if you put them side by side, a lot of people would have a very difficult time discerning which one was which. About 20, 30 years ago, this idea of xenotransplantation took place, where they were really interested in skin grafts and organ donation and whatnot. And it came down to the pig being the most useful model for doing that type of work. From a science side, we need to know the genome sequencing. There's been a lot of talk in the news kind of recently about trying to figure out the codes for the human genome. And mice have been completed for quite a while now. So we needed an animal model that had a complete genome sequencing. And luckily the pig was able to do that quite a while ago. And again, something near and dear to my heart, I've always been interested in the science behind reproduction, especially fertilization. So the pig really does serve a nice model for reproductive research, because again, it mimics that closely to the human. And there's still a lot of things out there that we don't know about. So basic benchtop research, if you will, is really focused on using the pig as that model. So that's why we picked the pig, because it made the most sense to what we were interested in. So we're kind of lucky today. One of my students happens to be here in the lab. The other two do have academic obligations, so they're learning. I think one's in genetics lab and one's actually in Spanish class. So luckily, someone didn't schedule classes this morning, so they're able to tell us a little bit about the basic day-to-day procedures we do here in our lab. So this is Kim, and she's going to walk you through some basic things.
Hello. So our process is split into 3 parts. The first part is oocyte maturation, then in vitro fertilization, and finally the culture of the embryos. Oocyte maturation takes about 2 days, and then after that we move into fertilization, which we do for about 6 hours. And after that's done, we culture the embryos for 7 days and observe them for changes. One of the problems that we have with in vitro fertilization is the high occurrence of polyspermic penetration in pigs, which is multiple sperm fertilizing the egg. In swine, this is about 30%, which isn't good because it results in the death of the egg. In the lab, in vitro fertilization rates are about 60%. And because of this, no in vitro system has been able to mimic the success of the in vivo environment. So we've found that everything in the in vitro system is linked. Typically, if we have a problem with the IVF step, it can be traced back to a problem that we have in maturation. And so it's similar in the embryo cultures. If we find a problem there, it can usually be linked back to the in vitro fertilization or the maturation. And of course, maturation creates its own problems if it goes wrong. We found that there are 2 different types of maturation in the egg. There's nuclear, which is the replication of DNA or mitosis. And then we've also found cytoplasmic, which is basically everything else that happens in the cell.
Okay. Again, as Julie said, I've been here about 10 years. And previously to that, I did some other work at another college over in Virginia, and then of course through the graduate program. But since 2010, since being here at the university, studying those different aspects of the system that Kim just talked about, we've been able to publish over 20 manuscripts detailing this and having over 70 presented abstracts throughout the world. We've gone anywhere from over to Paris, up to Canada, throughout our country, and then journals, again, worldwide. So it's been accepted by the scientific community. It's kind of like a little club, and we all share our results. And the thing that keeps coming back to us is not necessarily the science and what we've discovered, but the ability for us to be able to do this with undergraduates. And it's very difficult, and I have yet to find another school that's been able to have this type of discovery using just undergraduate directed research. So that's really the hallmark of our lab, is having this system set up, being able to explain it, and then letting the students pose a question and then try to solve it in their time here at the university. And in the beginning, it's a steep learning curve. They need to get the different techniques done. But by the end, I don't think I'd ever admit I'm sad to see them go, but they do leave and we have to start all over again. But I like to think that they leave their mark here because they do have published research and they are advancing the field. So that's really special, at least to myself. So again, we do run pretty much 5 to 7 days a week here in the research lab. Wednesdays are collection day for ovaries, and then we're pretty much here Thursday, Friday, parts of Saturday, sometimes Sunday evenings, and then back on Monday. Today is a Tuesday, so we are actually getting ready for ovaries tomorrow. So we really don't have a lot to show you, but we do, again, have some video footage that you can kind of see what we do that was recorded from a previous part of our marketing team. So we'll play that for you just so you can kind of see the overall operation. Hi, welcome to our lab. I'm glad you could make it. So we're just going to run through a couple things that we typically do almost every day of the week. The first thing we need to do is find a source for the eggs. So we go to a local slaughterhouse and get some reproductive tracts. So over here, we're starting to process the tracts by removing the ovaries. Skylar, how are the ovaries looking today?
They're looking really good. So good.
Good. And then once they're done, they're going to take the ovaries after they're washed. And we're going to go ahead and aspirate the follicles. So Skylar, what are you looking for?
I'm looking for follicles that are 3 to 6 millimeters in diameter.
So what's the easiest way to get those oocytes out of the follicles?
We take this needle and we'll go through and find the appropriate size follicles and aspirate them. And then the oocytes will be in the follicular fluid here.
So once we're done aspirating, we then transfer the contents of the syringe into a conical tube so we can let gravity do its work and settle the oocytes down to the bottom of the tube. Typically we go through 1 or 2 wash steps, which takes about 30 minutes. And then we'll spend a good amount of time searching for them under a microscope, looking for the various quality of eggs. So luckily, it's pretty exciting because we did this the other day, and we actually have some that have been maturing for at least a day or two. So if you'd like to get them out of the incubator, let's see what they look like. Thank you, Haley.
You're welcome.
So how long have you been doing research, Haley?
About 2.5 years. I started my sophomore year.
So these are pretty interesting. Again, it's been about 48 hours, and we typically put, I don't know, how many you guys put in a well typically?
About 50 oocytes.
About 50 oocytes, and you can kind of see the dark center is where the egg is, and then it has some cells around the outside that help protect it and give it nourishment and take away the waste products. So we like to see, we call it cumulus cells because it kind of looks like the big white puffy clouds you sometimes see when the weather's nicer. So this is something we really strive to get every time. What we typically do next is go ahead and take the cells off and then fertilize them, and then of course see what's going on and seeing if we can get some good embryonic development. Welcome to our. There we go. So again, since we really start we call it a cycle of production. So we start that every Wednesday, pretty much every week of the semester. Sometimes we start a week before the academic semester begins, maybe a week after as well. And then pretty much every other week during the summer too, depending on who's around. So we're pretty active. To get the numbers we need, we have to kind of go every week. And we're very, very lucky to have a really good source we work with up near the lake. So we've kind of been blessed with that ever since about 10 years ago. If we didn't have that, I'm not exactly sure what we'd be doing. So that's, again, what we do on a daily basis or pretty much a weekly basis. A lot of our background of what we've done previously. And then about 2 years ago, we started thinking about the next step. And we kind of gathered as a lab. We have much different schedules, so we really don't see each other all the time. And they're the students I have now are very self-supported, so they can actually, they do a lot of things without my help. It was a little heartbreaking at first, but when you know you're not needed, but at the same time, you're kind of proud of that. Anyhow, we started thinking about different issues in society and how we can maybe go beyond just some basic research and really trying to influence other aspects of the world. And it was very nebulous at first, but I'd like to share a little bit of data with you. This came out in 2017 by the USDA. And again, I'm going to guess most of you probably know this more firsthand than this poster does, but a lot of this is United States data too, that the cost of raising a child is extremely expensive. And a lot of goes into that, and it's very difficult to actually put a price tag on it. And as we've gone through the years, especially in the last 50 years, 40 years or so, the price keeps going up. And we still have lots of children. And in addition to this, let me see if I can get my laser pointer here. There we go. This doesn't actually even include the cost of a college education. So if the family, if the child makes the decision to go to college, then that's adding another price burden to it. So if the cost of raising a child goes up, and a lot of families choose to have more than one child, we get to exponentially increase that value as well. Take that into consideration. If you, again, from a little bit earlier data in 2012, the take-home message was they asked and they polled the public, what's the best idea or situation for the mother of that child, saying, well, should they be working full-time, part-time, or not at all? And for young children, I think it's pretty easy to see here that the ideal situation, again, according to most of the public, is that they should be working part-time or not working at all. And this is just personal preference. There's not really a lot of data to support their answers, but it's just their opinion. But regardless, the full-time working mother was very low on the percentages. We're not really going to address the male side of this today. But if you add the not working at all or part-time up together, you're looking at over 70% of either ideal for women or the young child is to spending more time at home. Now, take that into consideration with the previous slide of the cost of raising a child or children these days. It makes it a little bit more problematic. So in order for that to happen, again, the same study looked at whether or not the woman should be having children early in her career, later in the career, not at all. And if we focus down here at the bottom, I don't want to say an overwhelming majority, but a larger portion, if we put not having children at all or later in the career, is 61% compared to the 37%. So there's this trend of people wanting to have children later on once they understand and they can afford, for lack of a better term, to have that family. So based on that, you can probably see where this is going. Since the mid 70s, we've gone from the percent of women having children later on in life. We had a slight decrease, but it's starting to trend upwards again lately. Perhaps a little bit more interesting, at least from the science side of things, is over here on the right-hand side of your graph, is the mother's ages 40 to 44. Right, we're seeing a larger number of women deciding to have families, deciding to start families later on in life. and that's their decision. And again, through lots of discussion and through lots of exposure to media and whatnot, we know that's sometimes not the best idea for various reasons. So if we look at the mean or the average age of mothers in the United States at the first time their child was born, we're going from the early 20s, right, in 1980, which scarily enough is now 40 years ago, is, you know, below the age of 23, where 8 years ago, we're up almost to 26, and it's probably even higher nowadays. So there's very little argument against the idea that families or mothers, parents are waiting later in life to start their family. And again, the evidence was, well, maybe it's costing a lot to have those children, and they want to start their career and become established in their career first prior to starting that family, which is a lot different, so drastically different than prior to 1980. So where does that come into with our lab? So as the female gets older, there are more age-related reproductive capacity degenerations. And this is a natural phenomenon in mammalian females. As a mammalian female gets older, we see more issues reproductively. So a woman's fertility declines quickly after their mid-30s. And we're not really looking at trying to fix that. That's a biological process that we really can't change without massive manipulation. Early on, in the early 90s, two different groups, one in Europe and I believe one was from Australia, they actually took embryos out of women that were less than 35 years of age, and they transplanted them into surrogate mothers of older women, older than, I believe, 60 years. And those older women carried those babies, the offspring, to completion through pregnancy, through childbirth, and they had normal offspring. So what that told people is that it's not the individual, right? It's not the female, it's not the mother, it's not the person deciding to have children later on in life. It's not with them, it's actually with the oocyte, right? If we can get the oocyte or the female egg, the female gamete, right, the issue lies there. But looking and trying to figure out what's wrong with that oocyte is very problematic, right? Researching human oocytes, very problematic issue. So, and I think we can see where we're going here. Why not use the pig, right? Because remember at the very beginning of about 25 minutes ago, we said, hey, why use pigs? Because they're comparable to humans anatomically and physiologically. So what we decided to do, again, about 2 years ago, is use the pig as an experimental model and consider the aging process in that oocyte so that it can relate to humans, and specifically in our country, how women are waiting later to start a family and to alleviate perhaps some of those problematic issues that they might have. So we set out to create an oocyte aging model, right? We want to take this oocyte and in a normal environment, which we've hopefully shown that we've established quite a bit and been very successful at, and cause it to age. And we can't really just take the pig and let it live longer and, you know, keep it out in our front lawn and wait till it's, you know, 20 years old and then try to get the oocytes out of it. We need to do this in a lab. So our goal was to create an aged model. Once we did that, we can compare it to a normal model, right? The normal pig oocyte versus an aged pig oocyte. Again, relating it back to humans, creating an aged oocyte in a female, comparing it to, again, what we would typically call a normal model. Just being able to do that, of course, isn't enough. We wanted to measure key points, mechanisms, characteristics, the health of the oocyte through that entire process. Ultimately, of course, then, again, maybe you saw where this was coming, we want to test the ability to reverse that process. If we can create an aged model, then if we can somehow figure out what's going on and why it's going on, what if we can change it? What if we can say, okay, this is what happens to an aged oocyte. Let's prevent that from happening. And now we've eliminated or at least drastically reduced some of the issues associated with starting families later in life. So a couple, and now it's going to get, I don't want to say real sciencey, but a little more technical on our approach to this. So Kim was talking about the two types of events happening during oocyte maturation, and this is conserved across all mammalian species. So even though we're looking at it in the pig, it occurs everywhere. We see that in that nucleus, we have the DNA, of course, and I think everyone's familiar with DNA. The DNA is tightly packed into chromosomes, and in order to do so, it's wrapped around these proteins called histones. So if the DNA is going to be non-active and just hanging out, unless it starts to become activated, right? Fertilization, embryonic development, adults, once things are born, it's going to be pretty much in a state of inactivity. So it needs to be tightly packed together so it doesn't waste a lot of energy and it doesn't get damaged. So it wraps itself around these proteins called histones. Now these histones, here's that histone that the DNA is wrapped around. If we want to go through mitosis, if we want to go through fertilization, embryonic development, if we want the egg to be active like it should be, then we need to relax it, right? If you want to do something, right, you always have to relax yourself first. You don't want to go into something high strung. So in order to do that, the way that the DNA relaxes itself is we make changes to that histone. We attach what we call side groups to it. In this case, methyl CH3 is a side group we attach to it, the Me group, and we also attach acetyl groups to it, or the AC groups here. So the more groups that are attached to these histones, the more relaxed the DNA gets, and it can do things. We can make more genes, we can make more cells, we can develop an embryo, we can have a successful gestation, hopefully. So in order to, right, here's our histone, and the DNA is this, I guess it's a kind of a turquoise color. I'm not really that good with colors. But this would be an inactive segment of the DNA. If we acetylate it or acetylate it or methylate it, we're going to bind that carbon group or that acetyl group to these side parts of the histone, and it's going to become relaxed, right? And the DNA becomes loosely packed, and it can do various things, right? It can, what we call, get turned on. So the DNA, when it's turned on, it can actually go through cellular processes. Otherwise, it would not be turned on. It would be switched off when or prior to acetylation. How does this relate to the oocyte during oocyte maturation? Well, to go to an oocyte that is ready for the sperm to penetrate it or fertilize it, we need meiosis to occur, right? We need to take the total number of chromosomes in that cell and get it down to a single copy, because hopefully the sperm is going to provide that other copy. So we need to go through meiosis to reduce the number of chromosomes. That meiotic process is very difficult to follow. So it's a matter of turning the DNA on and off so that we can start to separate out the chromosomes and pull them apart and reduce that number. So that meiotic process involves a lot of acetylation and deacetylation. Right? If we want to move these chromosomes apart, we don't want them active. We want them just condensed so we can pull them or untangle them, if you will. So we need deacetylation to occur, right? We take the acetyl groups off, and now the DNA pretty much is inactive, so we can go through meiosis. So if we want to lengthen the age of that oocyte, we need to make sure that the DNA remains deacetylated. Turns out aged oocytes don't do that, and they stay relaxed, right? If we're holding this egg too long, it's like, oh, I'm just hanging out here. I need to start to do something or else I'm going to die or waste away. So it starts to naturally become acetylated. So in order to mimic aging, we need to get rid of, and this is a little confusing, the deacetylation process. And the way we do that is using a compound called trichostatin A, or TSA. So this is a deacetylase inhibitor. It stops the oocyte from remaining compact. It triggers the oocyte to continue aging. So we can take our normal setup, add TSA to the media, and then we get an oocyte that kind of is almost fast forwarded, like you go through commercials. I guess people don't really watch commercials anymore, but if you fast forward through them, right, you're accelerating the process or accelerating the age of that oocyte in a normal time frame. So we set up our aging model, right? We took our oocytes from the follicles on the ovary that we got from our slaughterhouse, and then we put them into a normal media, our control media without TSA, or we added TSA to it to accelerate that process. We would then, as again, if you remember, Kim said we go into maturation. So we put them in maturation for 24 hours. We wanted to lengthen that up. So we doubled that to 48 hours for a long treatment with or without the aging stimulant. So 48 hours with TSA would be an extremely long aging process. And then after that was done, we put them in the second half of maturation without hormones. Biologically, this is kind of, if you're familiar with the reproductive system, this is after ovulation. So we're mimicking as it's going down, I guess in humans it'd be the fallopian tube or the oviduct. So we're getting them ready for fertilization. And again, the other major part of this is to measure the quality of that oocyte after this process is done. So what we're doing is we're lengthening this maturation, and then we want to look at our quality points, as Kim said, IVF, embryo culture, consider nuclear maturation, that's the DNA, and of course, what else is going on in that oocyte. So we've come up with a lot of endpoints, oocyte quality. In the video, you heard us talk about cumulus cells. So a healthy oocyte has a lot of extracellular cells, the follicle cells that go with it after ovulation, expand after or while it's in maturation. So that's a good indication if that oocyte is high quality. We can measure if it's stressed or not using reactive oxygen species. That's the whole idea about antioxidants and taking on antioxidants in whatever form to reduce your reactive oxygen species in your body so you're healthier. We can just look at IVF, fertilization characteristics. If an oocyte is alive and viable and happy, if you will, it's going to be able to go through fertilization and, of course, embryonic development. We wanted to touch the nuclear maturation very simplistically. We're looking at meiosis. Where along meiosis are those oocytes going, whether they're aged or not? If an oocyte's not doing well, it can die multiple different ways, not to be morbid. But there's a programmed way the cells can be triggered to go through programmed cell death known as apoptosis. And we can look at various markers along the time points to see when this aged oocyte is now destined to not survive versus perhaps continue on to viable offspring. So again, we're kind of running out of time, but these are various measurements of apoptosis, whether it starts in the membrane of the cell, whether it's in the cytoplasm using enzymes called caspases, looking at the mitochondrial breakdown, and of course, looking at DNA breakdown. And then going back to those histones, right? We said the histones have to become acetylated or methylated so they can become relaxed to begin their process. And those are what we call epigenetic modifications. And epigenetics has, of course, been in the news a lot lately for various reasons, and how the environment can affect the DNA and then get passed down through generations, even though it's not strictly a genetic aspect. So we wanted to look at that too. So we looked at the process of methylation, which is called 5-MC. So it's again, if you're extremely interested in this, is where the methyl group attaches to the 5th carbon on the DNA. And then we looked at 2 different places where the acetylation would occur, on the lysine residue, on the 3rd histone, and then the 27th residue also on the 3rd histone. Those are 2 really nice markers that we've found that in that meiotic process of gametes or of eggs and sperm. So that's why we chose those 2. So again, over the last 2 years, we've created this model. We've looked at these endpoints to determine if our model actually does these things, and of course, for good or bad, for that matter. again, not to get too specific, but here's some data we've kind of put together in some of their presentations they've looked at. Again, we all know that recently everything's kind of been virtual. So we've had scientific conferences that are virtual. They do their presentations kind of like we're doing now, or they make a poster and then present their poster. So again, these have all been shown at various conferences. We've got the oocytes here on the left-hand side. We've used a green fluorescent tag to tag the reactive oxygen species. So the more fluorescence, the more stressed that cell is. So that would be this middle column. And then we just merge these two together using some software to get that fluorescence on the light microscope image. And again, didn't really label, just wanted to share with you some things we found. So down here at the bottom would be an aged oocyte, right? More fluorescence. Up at the top is kind of our control group, what you'd normally expect to see in a normal situation. So we're able, and we have evidence that we were able to create an aged model where we increase that stress of that oocyte. Didn't have any pictures for this, but just showing that we have been able to produce viable embryos in the stressed and lengthened model. So the TSA long had that trichostatin A added to it, and it was a long 48-hour maturation. We were able to get some viable embryos, but of course they are drastically reduced percentage-wise compared to our other treatment groups. Not to bore you with like lots of numbers, but these, and we can come back if anyone has specific questions about this, but it shows that we were able to stop and restart meiosis in that aging process. This is our apoptosis data, looking at the DNA breakdown. Annexin was the membrane breakdown or flipping. And then caspases is the enzyme reaction to that. And then of course the mitochondria breaking down, losing its membrane potential. So again, just some graphical data showing that we were able to increase apoptosis by the aging process and using TSA. And all these citations are the students in our lab. So these are all published by our students, which is kind of very cool. So that the mitochondrial matrix down here, we also did visually. We used two different fluorescent stains, light microscope image of the oocytes on the left. The red would be the mitochondrial membrane. The green would be the intact membrane. So we merged all three of these images together over on the right. And again, red is bad, green is good. So our aged oocyte, we had a lot more red fluorescence compared to the green, showing that the mitochondria are starting to break down in that oocyte, thus, again, an aged model. So to wrap things up, what's next, right? We still need to modify those epigenetic endpoints. And of course, we also have to start thinking about what we could test and discover to try to reverse that entire process. So that's probably going to be the next 2 to 3 years here as well. Unfortunately, with a new group of students, because they're graduating the next couple semesters, so that'll be also sad. But thank you so much. It's been a great time to share you through what we do. I'm of course obviously here to answer any questions you might have, or if you want to stop by, you're more than welcome to come visit the lab anytime you want. We always have visitors, and we might put you to work, but otherwise, hopefully everyone learned at least one thing today. So thank you.
Dr. Whitaker, I think everybody learned a lot of things today. That's a lot to take in, and it's so interesting. And I love how you're using, using swine to relate to humans in this way. So that's really unique. I had never heard of that until recently. And when I learned of your research, I just thought that it'd make a great presentation for us to learn more about it. I think that the University of Findlay has such a excellent animal science program. And I know it's broken down into a couple sections. So I'm hoping you can just chime in about this, and maybe even if Kim is still there, maybe she can even. She is
Great
good.
question for her.
So I see, so Kim, I'll direct this to you. So there's the industry option for animal science. That's one area of focus for the program. And then of course there's the pre-vet medicine, and then a science option. So can you tell me a little bit about the differences and tell me which one you're in?
So I'm a pre-vet major, and kind of the difference is the classes are more geared towards what your end goal is. So in the pre-vet track, we take a lot more chemistries and higher level sciences. And we also have a couple classes that are designated just for pre-vet majors that focus more on the medicine side of things. Like I'm in a class right now about food animal medicine, learning diseases and treatments for all of the livestock species. The industry classes are more tailored for people going into the management side of things, looking at finances and management of the farms, and then sciences is kind of overarching for everything.
Mm-hmm. Okay. I'm so impressed with the pre-vet program because I don't know if our viewers know this, but it has a tremendous acceptance rate into vet school. Over 85% from 2019 were admitted into vet schools across the country, which is more than 2 times the national average. So I think right here in Findlay, Ohio, we've got this program that is just exceptional, and that's so exciting. So Kim, tell us, what are your plans when you leave? How close are you to graduation? And then what do you expect to do?
I'm about a year away from graduation. I'm finishing up my junior year now, and my senior year is coming up pretty fast, but I plan to go to vet school after I graduate from Findlay, and I have started working on applications already.
Okay, good. Well, I know you'll do well. We had a couple of questions from the group. Joyce asks, is class size limited? So can you all, either Dr. Whitaker or Kim, can you speak to that and talk a little bit about class sizes? It's a general question, so I'm not sure exactly. Maybe you can give us a couple examples of class size limitations.
Some are and some aren't. It really depends on what the focus of that class is. Around here, we think of a large class. In reality, is probably not very big. We like to limit our freshman classes, at least in the animal science major, to about 35, and we think that's a large class. Of course, if you go to larger institutions, they're in the hundreds, so it's technically a really small class. As they progress through their academics here, the upper level classes have prereqs, which means you have to have certain other classes before you can take that one. So naturally, those classes kind of get smaller. So I teach, of course, the reproduction classes. So the sophomore level reproduction class is a required class for everyone in the major. And if we have 170 students, that's split over 2 semesters. So they typically run about 40 students in there. I also teach an advanced repro class, and they have to take the first repro to be able to get into that class. And that one's typically set at about 20. And it typically gets pretty close to full. So a lot of times we don't set them per se. It's kind of a natural sorting of the students on what their interests in. Mm-hmm. We have like production classes. They can pick any of our 5 major livestock species, and they get to pick, I think, 3. You have to take 3 production. Yeah. So 3 out of the 5 classes. So, obviously the pig one is not the popular one. So I end up with, some semesters is 15, whereas the sheep one and the horse one's typically at like 50 something. I'd say the beef cow, the cattle one is pretty high. So those students are more interested in that aspect. And so that we tend to get more students in those classes.
Okay, good. We had another question from Daniel and Donna, and they're asking, what kind of feedback do you get from your conferences concerning the research and the presentations?
We get constructive feedback. Just so everyone understands, so when you go to a scientific conference, they're typically about 4 days in length. Our students will go to other presentations, other poster sessions. They're typically on their feet. I know it's probably doesn't sound a lot to us, but for them, they don't like being on their feet for more than an hour, but they're pretty much on their feet from like 8 to 5, learning. And then if they when they do their presentation, it's typically either a 12-minute oral presentation with PowerPoint in front of an audience, or they're standing by their poster for about 2 hours. And they get the hundreds, if not maybe 1,000 people at this conference walking by, asking them questions. They get to go through their poster. And then it's not really judged or anything, but they're answering questions. And the nice thing about it is the people there actually want to learn about it. They're not trying to trick the students. They're not trying to ask the hard questions. They're really genuinely interested about their research. So the feedback's been pretty fascinating. Unfortunately, Kim's only been able to do this virtually, which, you know, you don't get a lot of feedback there. But in the in-person ones, a lot of the other institutions are impressed with what we do. We've been doing it for a while, so I don't want to say we're famous, but we're well known enough where 10 years ago, and no offense to anyone, it was like the number one question was, where's Findlay? Like, what state it is in, what country it's in. But now it's Dr. Whitaker and Findlay, and what are you guys doing this year, and that type of stuff. So just knowing that and getting over that geographic barrier, I think it's been pretty impressive, at least in my mind. But yeah, the feedback's been really strong, so it's good.
Good. I think that's just such a great experience. I'm sure Kim agrees, just to be able to present your information and then meet people in a normal non-COVID year. Where do these conferences take place? What cities?
Where are we going in December and January, Kim?
Pandemic allowing, we'll be going to St. Louis in December, and in January, Savannah, Georgia.
Nice, very nice. And do you see international students in the programs, and where do they come from? And then if that's the case, they go back with all of this great animal science perhaps to their countries, this great animal science experience and background and education. What do they do there? Can you tell us any examples from international students, Dr. Whitaker?
Like international students from university or from?
From the University of Findlay.
Oh, I'm sure they do. We do not have a large number of international students in our program. The ones we have had, I haven't taken them to conferences. I know they've been to a few, and they do go back to where they live and where they're from, and they start their careers. And I think it definitely helps them become more established and have a diverse array of experiences. And I think that even goes for the students that live here too. Any opportunity they have to go experience something else is worth it.
And to learn from others, to learn from their peers at these conferences, to find out what else is going on. It's just such a great experience.
Yeah, I think Mark Twain, I'm not sure if it was really Mark Twain, but he said international travel is the cure for ignorance or something like that. Right. So it's really nice so I kind of took that to heart, and I've been, all over the world doing presentations and whatnot, and having the students experience just something a little different, even if they're uncomfortable about it, it definitely helps them later on.
Yeah, good. All right. Well, Kim, thank you for being with us today. And Dr. Whitaker, thank you as well. That wraps up our Q&A session. And we're going to transition over. Stay with us, Kim and Brian. We're going to transition over, and we are going to spin the wheel for a prize. So I'm going to go ahead and share my screen, and we'll go over here. And we have a prize today. Our door prize is from Feasels here in Findlay on Bright Road. And so we like to support local businesses with our prizes. But if you happen to be tuning in from somewhere other than Findlay, let us know, because we are sensitive to the fact, and we're really pleased that this is a virtual event, and we've got lots of people attending from all over. So if you're not in the area and you're tuning in, let us know, and we'll work on a different prize. But Feasels Garden Center's got some beautiful things out there right now, and so we are going to spin the wheel and see what we can do to get a winner today. So let's spin it and see what happens. All right, our winner today is going to be Savannah Waters. And Savannah, we ask that you put something in the chat to let us know that you're here. So let us know. I'm going to open the chat too and see if I can see you respond. If you're not here, we're going to go on to another winner. So let us know if you're here. So far, I don't see anything from Savannah. Well, hold on. She had said earlier that she had lost her audio. Savannah, if you're still here, type in the chat and let us know. I know she was here, so that counts. And so we'll be in touch with Savannah. She may have dropped off because of her audio, but we'll be in touch with her and get that to her. And want to just take a moment again to thank our panelists for being here today. Don't forget, if you're returning for the 12:30 session, use a separate link. You'll need that separate link to come back and join us, but you won't want to miss the session. We are going to have a community update with alumna and Mayor Christina Murin. And joining the mayor will also be Crystal Whites from University of Findlay's Service and Community Engagement. She serves as the director of that department. She's going to be joined by students Nicole and Rachel, who will talk about their community involvement this past year. Want to thank all of you for a great lifelong learning month for April. Send us your feedback. We are interested to know what you liked and all of your comments. So email that to us if you can. And we want to hear that. We'll use that for future planning. Speaking of future planning, we're hoping to run lifelong learning in October in person on campus. We hope that we can do that given the pandemic. We'll keep an eye on it, but fingers crossed for that. We want to thank all of you for joining us today. And for all of you who have previously supported us, we thank you for your gifts, and your generosity ensures that we get to continue to offer this level of excellence that you deserve. So if you enjoyed today's event and you'd like to make a gift, please visit give.findlay.edu. And at this point, again, I'm going to say goodbye to Dr. Whitaker and all of his efforts and video, and Kim popping in to talk to us too. Many thanks to everybody, and we will see you all at 12:30. Bye, everyone.